I just want to start by thanking C&EN for selecting me and highlighting the work that we're doing, and thanking Stuart for a beautiful talk this morning. And so today I'm going to be talking about using environment-sensitive probes to diagnose tuberculosis at the point of care. All right, are you able to see my slides? So I will start with my origin story. I was born in Burundi, a small country in East Africa. When I was growing up over there, I quite honestly didn't know what my future would look like, and I certainly would not in a million years imagine that I would become a scientist. Back in Burundi, there are very few opportunities to do research for young girls like myself, and scientists did not look like me. Fortunately, when I was a teenager, I was afforded the ability to move to the United States to pursue the American dream, and that's where I was able to start my academic career. And so when I moved to the US, first of all, I didn't speak English. So I spent about some time trying to learn English and try to learn how things work here. And so after I was able to manage to learn a few things, I signed up to take some classes and think about what kind of degree I wanted to have. So first, I joined up at a local community college, and my very first semester at San Diego Community College, I took Introduction to Chemistry. And I was very fortunate that in that class, my teacher, Dr. Salwa Sadain, also spoke French. And so she helped me tremendously get over the language barrier and be able to excel in her class. And quite honestly, she became a mentor and a friend to me and showed me the opportunity that can happen in a scientific career. And it's through her mentorship that I actually was connected to the research community at San Diego Mesa College. This is Dr. Edward Alexander. He runs the Bridges to the Baccalaureate program at Mesa College, where he offers research opportunities to Mesa College students. So through him, I actually did a summer research program at University of California, San Diego with Dr. Rafi Oroyan, and I worked with him to do drug discovery for better treatment against intestinal worm disease. I was particularly attracted to his work because I understood the importance of the impact of these intestinal worm disease in places like Burundi, especially for young children. So with that exposure, I actually decided to transfer to UC San Diego and complete my undergraduate degree there in biochemistry. And so after a brief stint at UCSF as a summer research scholar, I worked with Dr. Christine Guthrie on mRNA processing. I became enamored with molecular biology, and that became my focus as an undergraduate. And when I came back to UC San Diego, I ended up working with Dr. Tracy Johnson, who is an RNA biologist, and we worked together on the mechanism of action and regulation of the spliceosome. And so it wasn't until I joined Tracy's lab that I really started questioning, or at least exploring, what my future looked like. For someone like me, before this time, I really hadn't seen a person that looked like me that was actually in academia and doing research. And it was through her mentorship, her friendship, and also serving as a role model for me that I was able to even imagine myself going to PhD programs. So I ended up applying to PhD programs, and luckily I managed to get myself a position at UC Berkeley in the molecular and cell biology program. So at Berkeley, that's where I discovered the beauty of chemical biology. And I think Carolyn Bertozzi needs a little introduction in this audience, but because I was so interested in research projects at the intersection of biology and chemistry, Carolyn's work in adapting or developing chemical tools to study biological systems was just spot on for me, and it was a bonus point that she also is an excellent mentor. So just to make sure that we're all on the same page, Carolyn coined, bioorthogonal chemistry. And so what is bioorthogonal chemistry? It just refers to chemical reactions that can occur in living systems without interfering with native biochemical processes. And so for me, this technology was incredibly powerful, and her and her team in her lab had done a lot of work to show what kinds of applications could be possible in the context, in the medical context. And so just briefly, what bioorthogonal chemistry really means is that you have your molecule of interest appended with some functional group, in this case, functional group X, that it gets inserted into a particular cell, and the molecule of interest is then exposed in the biological system, either inside the cell or on the cell surface. And then you can introduce a secondary molecule or a reporter molecule, either for fluorophore or an affinity tag, with a functional group Y that can selectively interact with functional group X to then load your reporter molecule onto your cell. So to me, this simple and elegant technology felt like it was open wide the opportunities that could be done in chemistry, especially in application infectious disease. And so I joined Carolyn's lab, and she had a project on tuberculosis. Again, because of my interest in global health and applications in infectious diseases, that's of course where my eye went. And so she had done, her and her lab members had already published on leveraging bioorthogonal chemistry to be able to introduce fluorophores onto the cell surface of mycobacterial cells. And so just a brief background, mycobacteria, Mycobacterium tuberculosis is part of the mycobacteria family. And this, to me, was important in the sense that we can then visualize mycobacteria and be able to monitor their viability. And so in this little schematic, we have a trehalose molecule that is a sugar base that becomes a trehalose glycolipid part of the cell wall. And in this example, the trehalose molecule has been appended with an azide group, therefore being azetotrehalose. And it gets inserted into the cell wall where it can then interact with a cyclooctine appended to a fluorophore in a bioorthogonal ligation step. And so then you have your fluorophore of interest exposed onto the cell surface that can be easily detected on a microscope. And so you can see on the images there, you see a bacteria, and under the green channel, you can detect the green fluorescence on the cell surface. And this process can work vice versa. So you could have alkyne group appended to trehalose, and then you're bringing in some azeto reporter group that can then also put the reporter onto the cell surface. And so this is what I would call two-step labeling procedure. So you have your primary molecule of interest with functional group A, and you bring a secondary molecule of interest with functional group B, and then you can tag your molecule of interest onto the cell surface for detection. There is a shortcut. You can also add just append a fluorescein directly onto trehalose, and then have trehalose fluorescein directly embedded into the cell wall, and you can detect the bacteria that way. So when I joined the lab, I was thinking about ways we can use this powerful technology for applications in the diagnostic space, and in a way that can also work in low resource environments. So what I thought would be really useful in this space would be to have a molecule that requires very little labor, technical skill set to use. So hopefully procedure that is very simple, that works really fast, a probe that is stable in different conditions, and is selective for mycobacteria. So we had the idea of using environment-sensitive probes to detect mycobacteria. And so here's an example of an environment-sensitive probe. So this is 4,4-N dimethylamino-naphthalamide, or short for DMN. And DMN is non-fluorescent in aqueous solutions, but highly fluorescent in hydrophobic environments. What we believe this meant is that if we were to attach DMN onto trehalose, then DMN will remain non-fluorescent up until it gets incorporated into the cell wall. So in this schematic, we have DMN trehalose, short as DMN tray. It gets inserted into the micromembrane, gets processed by an enzyme complex called antigen 5. And then once it gets inserted into the glycolipid layer or micromembrane, then DMN would turn on. It was a big gamble. We didn't know if this would work. And so we tried. So we synthesized DMN trehalose. We then did a quick first experiment to see if this would work. So the idea is that you have DMN tray at a concentration of 10 millimolar. You incubate DMN trehalose with mycobacteria for 30 minutes, and then you just image right away without doing any washing or any processing. And because of the simplicity of the protocol, all these steps would take less than 1 hour total. And so this is one of the first few images that we got. So here's a few mycobacteria cells, in this case, Mycobacterium smegmatis. And then I hope you can appreciate that you can only detect fluorescence from the bacteria itself. And just want to remind you that these bacteria are swimming in DMN, in solvent containing DMN trehalose. And so if we were to do the same exact procedure with fluorescein trehalose, you see that your image will be saturated by the extracellular fluorescein trehalose compared to DMN tray, the way you can actually just detect the cell surface DMN trehalose. So this was really exciting when we saw this working in this way. And for me, it was even more exciting because I knew that that was the first hurdle to get through. It had to be a simple process. And because we could directly detect and monitor labeling in real time, we could detect the labeling very quickly. And the earliest I could take an image was a 5-minute labeling. And this honestly was just the time it took me to add DMN and run to the microscopy room and then do the imaging. But since then we've done real-time imaging, and we can detect labeling as early as 30 seconds. And so overall, DMN showed us unique features that made it very attractive for a low-cost diagnostic reagent. You can do a no-wash, one-step staining procedure, very simple. It labels within minutes. It's very fast. And because it requires enzymatic action for the labeling step, it can report on cell viability. And so then the next question was, well, if it only labels live cells, then can we actually monitor drug efficacy with this? So then I did the same experiment, except I pre-treated samples with a drug cocktail, and then again added DMN tray, incubated, and imaged. And so in here, so we have 2 sets of images, bacteria that have not been pre-treated with a drug cocktail and a set of image from bacteria that have been pre-treated with a drug cocktail. So the cocktail includes the first 4-line antibiotics against tuberculosis, so isoniazid, rifampin, ethambutol, and pyrazinamide. And I hope you can appreciate in this image that cells that have not been pre-treated with drug are able to label just fine with DMN trehalose. But cells that have been pre-treated with a cocktail drug, therefore are dying or have died, are not able to label with DMN trehalose. This was really promising, and so we wanted to investigate a bit further. Well, if we can monitor cell death, can we monitor then drug resistance? And so in this case, we decided to label the cell, to pre-treat the cells with isoniazid. And the reason we chose isoniazid is because isoniazid is a prodrug, and that means it needs to be activated in order to be able to work against mycobacteria. And so this is a CatG-dependent enzymatic process. And so all we really needed to do is find a strain that it has deleted CatG. And so then we have a strain that is isoniazid susceptible, that is a wild type, and then a strain that is isoniazid resistant, that is a strain that does not have CatG. And so in this image, I'm showing you the mean fluorescence intensity of a collective of cells that have been labeled with DMN trihaloase. And each of these samples have been labeled with increasing concentration of isoniazid. So for the wild type, as you increase isoniazid concentration, you're able to monitor a reduction of DMN3 fluorescence. And that makes sense because cells are dying. But for isoniazid resistant strain, despite the increasing concentration, despite the presence and increasing concentration of isoniazid, we did not see any reduction of DMN3 labeling, suggesting that the cells are resistant to isoniazid, and therefore they're not dying, and therefore they're labeling just fine with DMN trihaloase. This was really exciting for us because for me, moving forward, I saw that not only can DMN trihaloase serve as a biomarker for the rapid and simple detection of TB, but also because there's a potential use for treatment efficacy monitoring. So why do I care so much about TB? TB is the #1 lethal disease that is caused by a single pathogen. I mean, everyone now is sensitized to infectious diseases because of coronavirus. But TB has plagued the world for centuries. And as you can appreciate in this image, my country, Burundi, has a lot of TB cases, and it's one of the things that actually drive mortality in Burundi. So despite being a preventable and curable disease, as we know, TB remains a deadly disease. About 1.5 million people died of TB in 2018, and about 10 million people get sick every year. And 30% of those cases, about a third of those cases, are missed due to lack of point-of-care resources. And the major problem is that people are in remote places. They do not have access to clinics with fancy equipment. And so the cases are missed. And so for me, I understood that rapid screening of TB at the point of care could save millions of lives. And so for that, I wanted to just directly get into the clinical aspect and look at whether DMN trihaloase can actually detect TB in clinical context. So I flew over to South Africa and decided to connect with Professor Bhavesh Khanna at Witwatersrand University in Johannesburg, South Africa. And I collaborated with him and Christopher Elan, his postdoc, to use DMN trihaloase and detect TB, Mycobacterium tuberculosis, in sputum samples from TB patients. So the process is simple. The goal was that we would collect sputum from patients with TB who have been diagnosed with TB in clinics, do a simple decontamination step so that we can work with the nice samples. If anyone has ever worked with sputum, they understand this problem. And once we have a workable sample, then we can either do DMN tray labeling, as I've explained before, or perform the standard Oramine smear test, which is also a fluorescent staining procedure. And then after that, we do the imaging. And so here's an example of what we saw. So I'm again showing you two sets of images. On top is DMN tray bacteria that have been able to be found in sputum samples, and at the bottom are Oramine stained smear test. And I hope you can appreciate that not only can you detect TB cells in sputum samples, but also Oramine is a 7-step, 2-hour procedure, and DMN tray is a 1-step, 1-hour procedure. It's just a lot simpler to use DMN Trihalo's in this context. And so we have done a lot of work since then, but I hope that I have shown you that environment-sensitive biomarkers provide many benefits. Just with this example with DMN Trihalo's, I've shown you that we can follow a no-wash 1-step staining procedure that is easily applicable in low-resource settings. DMN Trihalo's can label within minutes and therefore makes the detection very fast. And importantly, because DMN Trihalo's requires enzymatic processing, then DMN labels only live cells and therefore can report on drug efficacy. What was most important for me was that DMN Trihalo's uses existing equipment. I was not asking clinicians in Johannesburg to get this fancy new machine. I was not asking them to use different materials. This is just a simple test that they could do, they were able to do by themselves on their own with a simple procedure. And so because we understand the potential for DMN tray and other environment-sensitive biomarkers in this context, we decided to form a company, a public benefit company called Olilux Biosciences. And so at Olilux, we are driven and motivated to build these environment-sensitive probes for a wide variety of application. Right here, this was just an example for TB diagnostics, but you can imagine you can also you can monitor drug efficacy for TB. You can append these environment-sensitive probes on wide-ranging molecules of interest and for many other applications in research and medicine. A particular project that I'm really excited about is the potential for mobile apps and telemedicine using a kit that we're developing with DMN Trihalo's and a portable fluorescence reader. So the fluorescence reader is a device that will complement DMN tray staining. It is battery-powered, portable, and low cost. And importantly, because of the new technology advances that we've done, we can also have a reader that can connect remotely. And as a result, we have this device that we can put in a backpack and we can go find TB patients where they are and therefore do TB screening. So with that, I will thank everyone who's listening to me. I will have to thank Carolyn and Carolyn's lab, all the collaborators, Bhavesh, Manu, and Prakash, and all my trainees that have helped me all in this project. I always joke that I had a mini lab in Carolyn's lab, and I'm so thankful and grateful for all the work and the support that I've enjoyed thus far. And I thank you for listening.
Wonderful. Thank you so much, Mireille. I think you can hear me. I can't see there I am. Thank you so much for the great talk. We're running really tight on time, and I hate to say that I'm going to skip the Q&A. There are some questions in the Q&A box. I'll ask if maybe Mireille can take a look, or if there's time at the end, we'll get to those. Thank you so much. That was fascinating.